Good morning, respected judges and panel members. We are TechMed 6, presenting our proposed solution under problem statement 25042, category software. Our project addresses a critical gap in marine genomics, the massive proportion of unassigned environmental DNA sequences from deep sea samples. Currently, over 70 to 90% of deep sea eDNA data remains unclassified, constituting what we refer to as genomic dark matter. Our objective is to eliminate this dark matter through a novel AI driven pipeline that integrates environmental DNA analysis with machine learning, providing unprecedented insights into deep sea biodiversity. Our proposed solution, titled Deep Taxa, integrates artificial intelligence with environmental DNA analysis to advance deep sea biodiversity discovery. The process begins by collecting eDNA from deep sea water and sediment samples. Our AI driven pipeline then cleans, extracts, and clusters DNA sequences to uncover meaningful biological patterns. This system not only identifies known species but also flags novel taxonomic clusters, enabling the detection of previously unknown organisms. The core innovation lies in three aspects. Number one, tackling genomic dark matter. We employ deep learning techniques, especially variational autoencoders, also known as VAE, combined with clustering to identifying unique genetic patterns without relying on existing reference databases. This allows our system to interpret unassigned sequences as potential new taxa, effectively revealing hidden biodiversity. Overcoming the alignment bottleneck. Traditional alignment based searches are computationally expensive and slow. Deep taxa replaces these with direct pattern recognition and clustering, significantly reducing computational time and enabling faster diversity assessments. Number 3, breaking discovery bias. Our models detect and prioritize novel or divergent sequences that traditional tools often filter out. This approach broadens discovery beyond a known species, providing a more complete ecological perspective of deep sea environments. As illustrated in the performance comparison, our solution achieves a dramatic increase in identified species while minimizing the percentage of unassigned sequences, demonstrating superior detection sensitivity and analytical efficiency. In summary, Deep Taxa represents a first of its kind end to end AI pipeline that goes beyond known biodiversity, uncovering new life forms and supporting data driven marine conservation.
System architecture and workflow. Here the workflow is divided into four major phases. Phase 1, data preparation. We import raw, FASTA or FASTQ reads, perform adapter trimming, low quality filtering and dereplication to obtain clean, non-redundant sequences. Now phase 2, that is AI core feature learning. The sequences are numerically encoded as k-mers or one-hot tensors and passed through self-supervised neural encoders. The models learn compositional and contextual patterns without requiring labels, producing dense embeddings sensitive to even rare species variations. Now phase 3, that is analysis and interpretation. We employ clustering algorithms such as k-means and HDB scan to define operational taxonomic units. Novel clusters without database matches are flagged for further biological investigation. Now phase 4, that is the output and visualization. Visual analytics tools include TSNE, UMAP and phylogenetic trees, present abundance metrics, evolutionary contexts and biodiversity indices.
Now we come down to slide 4, our technical readiness and feasibility. Our AI components utilize proven high readiness architectures such as implemented via PyTorch, TensorFlow and Scikit-learn. Importantly, no label data is required due to self-supervised learning, and existing CMLRE datasets of the deep sea eDNA can be directly integrated. The system operates efficiently on standard GPU clusters, replacing slow alignment methods with rapid interference pipelines. Training is a one time computational cost, after which interference is fast, scalable, and cost effective. Now we come down to slide 5, your innovation and scientific impact. Our model effectively addresses the largest bottleneck of the deep sea metagenomics, the inability to classify unassigned sequences. It converts discarded reads into biological meaningful data, improving species detection sensitivity by a factor of 2 compared to conventional workflows. Through graph neural networks, novel sequences are linked to phylogenetic lineages, generating traceable hypotheses that accelerate scientific discovery. This transforms raw genomic data into context rich diversity intelligence.
Applications and benefits. The proposed pipeline supports multiple high impact domains: evidence based conservation, generating high resolution biodiversity baselines essential for marine protected area and vulnerable marine ecosystem assessments, rapid environmental monitoring, near real time eDNA analysis following mining or pollution events, enabling swift ecological response, bioprospecting, identifying novel organisms with potential industrial or pharmaceutical applications, policy and compliance, facilitating data driven decisions for the blue economy through integration with OBIS and other global biodiversity systems. In essence, we provide a standardized AI driven framework that converts raw deep sea DNA data into actionable insights for conservation, regulation, and research.
To conclude, the TechMed 6 AI eDNA pipeline represents a significant advancement in marine biodiversity discovery. By emerging artificial intelligence, self supervised genome, and graph based interfaces, we overcome the limitations of traditional taxonomy and extend humanity's reach into the unexplored biosphere. Our approach transfers eDNA, once considered waste, into a scientific resource of immense ecological and economic value. In doing so, we position CMLRE and India at the forefront of AI based deep sea research and sustainable ocean management. Thank you.